And I'm pleased to introduce the first speaker of this session, Astrid Graslund from Stockholm University, Sweden, and she will talk about biophysical studies of metal-ion interactions of the A-beta peptide, modulation of the amyloid aggregation process. Please, Astrid, the stage is for you.
Thank you. Well, I would like to start by thanking RAMS for organizing this meeting. It looks to be very interesting. I am happy to share some of our results with the rest of you. And I realize that many of you are actually experts on this field. But I will start by just reminding you of the amyloid beta peptide. I have indicated in red here the histidines in the sequence in the N-terminus. And as you all know, it's 40 or 42 residues long. We have certainly over the years studied many metal ions and seen that they all seem to interact and bind more or less specifically with this peptide. So, of course, the important question at the end, which we will not probably solve today, is what is this relation between the metal binding and the disease, the Alzheimer's disease? So, we have a model here that we draw some almost 10 years ago, where you see and you recognize, of course, the monomers, the hairpins, which are supposed to be part of the structure in the end in the fibril, but we also have disordered aggregates and so on. So this is a rather, you could say, wild scheme. And our problem in many of our experiments is, of course, to try to stop or just to look at a particular stage of this scheme. As you all know, probably we nowadays think that it is somewhere in this region before the mature fibril is formed that we have the most toxic state of the peptide when it is partly aggregated. So we did some more models some years ago, and just to show you that we have structure models up here, we have AFM images down here, and we have different different stages of chemical exchange. So we think there are similar binding sites at the N-terminus for both metals and metals, and we could also see increased rigidity and so on after zinc binding. So we also looked at what are the ligands, and we could verify that it was histidines, and it was actually the N-terminus. So it is for ligands and not tyrosine 10. And this was what we know more than 10 years ago. And we could even draw a model of zinc binding here, and it looks similar for both copper and zinc. And we have a folding around the metal ion in the middle, as you can see. So at thioflavin T, of course, we did experiments to do kinetics of amyloid formation. And we could do this rather carefully by A-beta alone at different concentrations. And then we would add more and more zinc and then see what happens to the kinetics of the amyloid formation as measured by thioflavin T fluorescence. And here we could see, yes, we could measure it in many different conditions. And the final observation is that yes, zinc retards A-beta fibril formation as measured by thioflavin T, and we could also see that they were the same aggregation mechanisms with and without zinc, because you could just let them go one after the other. And this is again relatively long ago, but we think we knew quite well about what happened with zinc. And then we, of course, in the meantime, we have had theoretical work from the people in Cambridge, Knowles and company, so to speak. So we can talk about primary pathways, elongation, secondary pathways, fragmentation, surface catalyzed nucleation, and try to figure out where is our metal doing something. And there are different exponents in the curves that you can draw for various variables. And in the end, we could see that zinc binds specifically to this N-terminus ligands, and it retards the process of aggregation by fibril end perturbation, because it can't end. It can't add another peptide when you have a metal doing something in between. And that, of course, means that you have slower aggregation because you have a disturbance of less active monomers, so to speak, that can add. So with that, we went on and we studied also a beta 40 bound to a micelle. That is the simplest way of letting a membrane mimetic go into the stage here, and zinc was bound also here, we could see, and we have formed alpha helices binding to the micelle, which we found quite interesting. These are the parts that will become in the beta hairpin, will have beta structure. Here it's alpha. And we could here look at metal binding again to micelle bound, and we had very similar ligands in the N-terminal segment of both copper and zinc when the peptide was bound to this SDS micelle. So if that is a reasonable model for a membrane, a proper phospholipid membrane with head groups, then we can see that yes, the N-terminus seems to be outside of the membrane, and it can add metals to it and they can do something. So we would also be able to see that these metals, the redox active metals like copper ions here, could actually lead to production of hydrogen peroxide, which here we take as an example of reactive oxygen species. So it doesn't matter if you have SDS present or if you just have A beta and copper present. So this we believe is, we come to that at the end as a model for possible toxic mechanisms. And so we would conclude about the A beta peptide that we have an in vitro aggregation process which we can follow also actually by CD. I didn't show you that. We have metal ions that interact and they affect how the kinetics appear. And we also can see reactive oxygen species in such systems. So then we have other metal ions which can be involved here. And that would be, for instance, manganese, which we have studied more recently. And that seems to bind more or less in the same region, a little bit less specific, and that seems to be the difference between different metal ions. They all are in the same region, but more or less specific, and of course more or less strongly in binding under certain conditions. But again, you see here that manganese, which is also redox active, will make all the signals in the N-terminal residues go down tremendously. Again, we have looked at silver ions, which are 1+ ions, and also they are actually bound in the same region. And again, we have a fibril end elongation, which seems to be the mechanism by which they retard the fibril formation. And I should also mention here that of course we have always substoichiometric amounts of these metal ions. If you have very large concentrations of metal ions, like you talk rather about ionic strength instead, then of course you have completely different observations. Then you can actually make the reaction to form amyloid much quicker because you reduce the electrostatic interactions. But here again, we have available pool of monomers decreased and we have retarded aggregation. So I'm coming close to my end here. we see that the histidines seem to be involved in all the metal binding that we have looked at. We have compact folding in the N-terminus. We have actually millisecond exchange time. I didn't go into that, but we can measure that by our methods. And we see that fibril end elongation is retarded. So then we have a speculation. Does binding of redox active metal ions to A-beta cause oxidative damage, for instance, in a cellular membrane? And that we are doing some things now to actually look at. And I believe that this is the case, that this is one of the toxic mechanisms. Sorry, I should, why the A-beta peptide is toxic to cells in the brain. And then, of course, I did not go into what we have also looked at. We see peptide dimer formation via dityrosine formation, and that also affects how the amyloid aggregation occurs. So there are still many questions unsolved, but I think we are fairly good, I would say, in our understanding of how these more common metal ions interact with A-beta in the monomeric form. Absolutely different, you can expect to see things when you look at the oligomeric states, if you can stabilize them. And then, so I'm not speaking too long, even though we had some problems to begin with. I want to acknowledge my co-workers here at Stockholm University. And Cecilia did a lot of this work. She has finished her PhD. Niklas is another PhD student who has another year to go. And co-workers, more senior ones, are Jyri Järvet, Sebastian Wärmländer, and also we have others, neighboring departments like Jens Danielsson, Leopold, Ilag, Sabrina Scholz. Jens actually is at our department, but in another group now. At the Karolinska Institute, we have Vladana Vukojevic, and I have not gone into that work where we do fluorescence correlation spectroscopy, which is another topic here, but not including metal ions so much. And then, of course, we have collaborators in different other countries which have been more or less involved in the metal binding work here. And I see that Birgit Schroeder is here, and you will talk about other aspects of metal ions. So I think with that, I will stop, not to be too long, and then I'll be happy to take questions. So do I do stop share now, I suppose?
Thank you, Astrid. No, it is not necessary to stop sharing now, because we have to see whether there are some people from the floor that can type some question.
Okay, I see.
I cannot see any question in the box. However, I don't know if it is a problem of mine of.
Yeah, I have chat 1, it says on my screen here. What does that mean? That there is a no,
I don't see,
unfortunately, 0. Yes.
I don't see any question in my box. I'm sorry, so I cannot read it.
Too bad. We are.
I have, okay. I can see just one. Okay, I just see one question from Sahil Halawat. Sorry. Okay, the question is: zinc usually have 4 binding. I'm sorry, it disappeared. Okay, zinc usually has 4 binding sites. Which is the 4th residue involved?
Yes, a good question. And I mentioned it in passing. I said the N-terminus, but it could actually be also the side chain of aspartic acid 1, which is there. And it varies a little bit for different metal ions, and it is more or less specific. So I wouldn't say, whereas the histidines for zinc are quite specific, the 4th ligand is not quite specific. But if you looked or saw my model, it was the actual N-terminus that I had there. I should also say that zinc may have a second binding site around residues 23, 24, 25, weaker binding there, which we have not really pursued. But it's a good question. And this varies between different metal ions, actually. The histidines seem always to be there, but the fourth one can vary.
Thank you, Astrid. We have other two questions from Lothar Bremer. I see in the chat. His question is, in some fibril structures, the N-terminus is free, including the histidines. Can you comment on that?
Yes.
For example, on the inhibition of fibril formation by example, give a copper or zinc. This is the first question.
Well, that's another good question. Yes, I think that in many of these, you could say, aggregates or fibrils, you have free N-termini and they can bind metal ions. I'm sorry to say that we cannot really answer what do they do, but they will certainly affect the secondary nucleation, for instance, one way or the other. And it's, I would say, very unfortunate that we have these three histidines sitting there and trying to bind metal ions. That's how we have amyloid formed by the peptide, I would say. But and the fact that they also bind and are visible on the fibrils show that also there you can have metal ion effects. Yes, and if the fibril, for instance, sits on the membrane, and then you have the metals are then, of course, brought very close to that membrane, they can undergo reactions that may damage the membrane. Yes.
Okay, Astrid, thank you. Next question is from Feng Ding. What are the binding affinity of metals with A-beta fibrils? Does metal binding affect the fibril structures?
Yeah, that's a good question. And I cannot say. We have only measured properly metal binding affinities to monomers, not to fibrils. So I could not give you a good answer. And I cannot even say how will the metal ions affect the structures of the fibrils. Maybe not at all, because the real structures of the fibrils are made not from the N-termini usually, but from further down the sequence. So it's a good question, but I don't have a precise answer. Maybe some of the other speakers that will follow after me will have better knowledge about these things.
Okay, we have another question from Candace Strang. When do you think micromolar free zinc is present in the tissue? Most sites are nanomolar, so this site will not compete for transfer of bound zinc.
That is true, but I think that there are some parts of our bodies or some parts in the brain where the metal ion concentration is more obvious. It's where you have, I don't know the proper names in English, but where the neurons interact with one another, you may have higher concentrations. But but I do agree that what we have to study is, of course, higher concentrations, because we have these other methods that need reasonably high concentration of the peptides, something like 5 or 10 micromolar, and also micromolar metal ion concentrations. Yes.
Okay, next we have another question from Sikun Liu. Thank you so much for the great talk. Have you looked at A-beta 42 apart from A-beta 40?
Question is yes, but I couldn't show you all our results, so to speak. So we have done that, and in almost all questions where we have looked, of course, we usually have lower concentrations of A-beta 42 to have a monomeric state, but it seems to me that the binding properties are similar, and certainly the binding sites and the roles of histidines in the N-terminus seems to hold up in the similar way.
Okay, I think that we have time for the last question from Sahil Halawat. Are the fibrils formed in the presence of zinc homogeneous?
Yes and no. I mean, no fibrils are completely homogeneous. And you have fragmentation and you have secondary nucleation and so on. As far as we have been able to see them by EM, I think they look very similar, yes. But one should remember that all fibrils formed by amyloid beta peptides, whether 40 or 42, are not quite homogeneous. They are heterogeneous and they are different sizes and certain different properties.
Okay, thank you so much, Astrid, for your very interesting talk.